Good afternoon, everyone. So, as you have already understood that today's topic of discussion here will be on the different tests, on the biochemical test on protein, and these kind of tests will be quantitative tests. And today I am going to show you all this test along with Debashish sir, and so here it goes. So the first test that we are going to perform is known as the Heller's test. Heller's test is a very simple and easy kind of a test. In this case, we will be using the reagent, that is nitric acid, that will be the primary reagent here, and we will be taking the solution in a sample and we will be pouring down the nitric acid through the wall of the test tube slowly, so that there is a distinct kind of layer, two different layers can occur, and let's see what we can find in the result of this test. Stay tuned. So the first step of this reaction is to pipette out some amount of nitric acid. So here in the test tube we have a sample solution. Can we see the sample solution? Okay, so we can see the sample solution present here, and now we are going to pour some amount of nitric acid. So this is nitric acid. So this is a concentrated nitric acid, right? Okay, so this is a concentrated nitric acid. So Debashish sir is using the scroller system, and he took some amount of nitric acid, a few drops of nitric acid, and after that he is adding this directly to the sample. Please note that this sample has to be kept in a slanted test tube. So can we see this result? Yes. So I think you can now see that as soon as the nitric acid has been added, there is a small amount of precipitate, white precipitate, that is occurring right here. So this is one of the major tests that we can see. We can say this is a confirmatory test for protein. So with this test we can say that protein is present in a sample. This same test we are doing with a blank. By blank we mean that there is no certain sample is present. So that is plain and simple water, and he has added some amount of nitric acid to water, and as we can see that there is no precipitation which has been formed. So this is one test we can confirmably say that whether a protein is present or not. So the next test that we are going to perform is known as the biuret test. For the biuret test we are going to require two major type of reagents. The first reagent that we require is called the sodium hydroxide, which is a highly acidic solution. The second thing that we require is copper sulfate. Copper sulfate, we know it's a copper salt, and let's now see that what is the result that is coming out of this test. So for this purpose what we have done is that we have created a concentrated sodium hydroxide solution. Around 40% weight by volume solution has been created, and the second thing is that copper sulfate, which is also known as the cupric sulfate. Extra pure means, the term extra pure means that there is a high amount of purity of cupric sulfate. So this solution, as you can see, this is a blue solution that has been created. Now what he is doing is that he is adding the sodium hydroxide solution. Please add the sodium hydroxide solution. Okay, some sodium hydroxide solution has been added now, and some copper sulfate solution is now being added dropwise. See, we can see there are some kind of changes which is occurring, and as you can see that there is a brilliant kind of purple color which has occurred. Please remember that this purple color is actually the confirmatory test of a biuret test. So I think now you can understand that what will happen if we add first concentrated sodium hydroxide solution and then some amount of copper sulfate solution in protein sample. Now let's see what happens if we do the same thing in case of. So now what we are doing, we are using simple plain water, and thus we are testing on a blank. Please add the sodium hydroxide solution first. So the sodium hydroxide solution is now being added, and after that, drop wise, he is now adding some amount of copper sulfate. So after the addition of copper sulfate, we see that there is no special kind of change, and the color which is coming here is the color of the copper sulfate itself, and no changes are there. The next test that we are going to perform is known as the saturation test. Now saturation tests are of two different types. The first one is known as the half saturation test. The second one is known as the full saturation test. Now in case of both half saturation and full saturation test, there is one single type of reagent that we are going to use, and that reagent is ammonium sulfate. For half saturation test, we will be using a solution of ammonium sulfate, whereas in case of full saturation test, we will be using the solid ammonium sulfate crystals. Now let us see what are the different type of changes that will occur when we add these type of solutions or reagents in a case of a blank and in case of a sample. Now we are going to perform the half saturation test. So in this case, what we have, this is a pre-made solution of ammonium sulfate. I think you can see this here also. It is written ammonium sulfate. Now ammonium sulfate is now being added to the solution. So carefully he is adding some amount of solution. As of now, there is no change, and let's see if there is some kind of change that can occur. Okay. So the next thing that we are going to do is full saturation test, where we are adding ammonium sulfate crystals directly to the solution, and let's see what happens after the mixture. What happens in sometimes that these kind of reactions, as these are biochemical reactions, they might take some time to go to its final result, and let's see if there is some kind of change. So if we check the full saturation test, we can see that there are some precipitation which has occurred, but in case of half saturation test, though there is no precipitation has occurred, there is slight amount of change in the concentration. As we can see, it is now a little bit of cloudy. So we can say that this test has been confirmed. Now the same reaction is being repeated in case of a blank. As a blank, we have just water present. Now he is now adding some amount of ammonium sulfate solution. As this is water, see the water remains as it was. There is no change in the transparency. And the same thing is happening for the full saturation, where we saw that there is a huge amount of precipitation. Here he is also adding some amount of ammonium sulfate, as you can see here. As soon as he added some amount of ammonium sulfate, there is again no change. So no change is there as expected. The next test is known as the sulfosalicylic acid test. For sulfosalicylic acid test, the reagent itself is in the name. We will be using a sulfosalicylic acid solution. Now we need to add the sulfosalicylic acid solution to the sample drop by drop, and we need to mix it properly. Let's see what will happen when we are performing this sulfosalicylic acid test. So this time we are going to perform the sulfosalicylic acid test. Now this is the protein sample that we have taken here, and this is the sulfosalicylic acid solution. Now let's see what will happen. So he is now adding it drop by drop. Please remember that it has to be added drop wise, and after that it has to be shaken vigorously. If we observe it very closely, we can already see that some kind of precipitation, and we do not need to observe very closely, rather it is already present there that how vigorously the precipitation has occurred here. Now we will now check this one with the blank. Now as a blank we have taken water, and let's see what happens if we repeat the same experiment with water. Now he has added a few drops of sulfur-salicylic acid in water. I think that will be enough, and as we know that there is no such changes. Next, we are going to perform something known as the lead sulfide test. For lead sulfide test, generally there are two different type of reagents which is given. First is the 40% concentrated sodium hydroxide solution, whereas on the other hand we will be giving a solution of lead acetate. Now lead acetate will be reacting with the sulfur which may be present in case of any protein, and then it will bond together to form the lead sulfide. So that is why this test is known as the lead sulfide test. Let's see what will be the result of the lead sulfide test. First, now we have taken a sample in the test tube. As this experiment will require some amount of heating, Devash is now using a special type of apparatus which is known as a test tube holder, so that the heat does not get transferred into his hand. So now he is adding some amount of sodium hydroxide. Now he is adding it drop by drop. Around 2 ml of sodium hydroxide has been added. Now what we are going to do is to boil it. Now we need to do it for a minute. I have paused the video for some time. Now we have passed 1 minute, and now we can already see that in the water bath which was already on, it is now boiling. Now after this one, what he is doing, he is adding some amount of lead acetate. Now let's see what happens now. Now as soon as he added some amount of lead acetate, there is a huge amount of precipitate which has occurred. So this precipitate, now we need to know that whether there is a brown or black type of precipitate. Now yes, as he is showing to us that there is a strict change into the color, and the color is now going brown. So we can now say that this type of test has been confirmed for sulfide. The next and the final test that we are going to perform today is known as the xanthoproteic test. Now the word xantho itself means yellow color. So obviously it is very easy for us to understand the end product will actually have a yellowish color. What are the reagents that are required in this case? The reagents that we will require is first of all nitric acid, and after that we will require some amount of sodium hydroxide. Now in both the cases, after the reaction, at first we need to heat this up for a few minutes, and there will be precipitate, as just like what we have seen in the Heller's reaction. There will be a precipitate that can be found, and after that we need to boil it. We need to boil it for around 1 minute, and after the boiling process, what we can see is that the resultant substance is then further reacted with sodium hydroxide. When it is reacted with sodium hydroxide, if there is any kind of yellow color present, the result will be into xanthoproteic complex. Always remember that this kind of experiment tells you that whether any aromatic hydrocarbon or aromatic ring or aromatic amino acid is present in that particular kind of a sample. First thing first, we need to add some amount of nitric acid. So some nitric acid is now being added. Okay, if we see it properly, there is already a precipitation. Now let's see what happens after the boiling process. Now we are putting it into the water bath for boiling purpose. Now after the boiling process is over, we are adding some amount of sodium hydroxide. What happens in this case? If we shake it properly, yes, we have the yellowish color which is required. Now of course this is a protein solution which we were using, and there will be some amount of amino acid containing aromatic group containing amino acid. So this is the result of the xanthoproteic test. Now we have all the tests which are present in front of us. The first one, which is already present in the left, is the first test, and that is known as the Heller's test. The next one that we have performed is known as the biuret test. After biuret test, there is full saturation test that we have performed. The next one is known as the lead acetate test. See, we have the brown color, and how the brown color is now deepening with the time. The next test is known as the xanthoproteic test, which actually tells us about whether any kind of aromatic acid is present. And finally, what we have is known as the sulfosalicylic acid. So these were the basic tests of different type of proteins, and I hope you have understood about these experiments, and if you have any questions, please put it forward. Thank you and have a good day.